Hello and welcome to our open house here in the Cancer Center, Sanford Burnham Prebys Medical Discovery Institute. We welcome you, and I would prefer to welcome you in person in our Chairman Hall. Hope to do so in the next time. Today, the virtual tour that you're going to attend will take you into our more recent discovery in cancer and coronavirus. I am Zev Ronai. I'm a scientist, professor here at the Institute for the last 15 years, currently the interim director of the NCI-designated Cancer Center. Our cancer center is one of the few basic cancer centers in the nation, and the work that we do here focus on addressing critical problems in cancer, fundamental problems that help understand and solve unmet clinical needs. The unique thing is that we do it here in collaboration with a number of our own folks, about 40 of us working on Cancer Center, collectively with a number of core facilities that specialize in drug discovery. What you're going to hear today is about work that was done recently in immunology, in cancer research, in coronavirus. The COVID-19 has impacted us all at number of levels, and soon, or relatively soon after we were closed down in mid-March, we started to come back and work on, some of us that were allowed to come back, work on coronavirus problems. And that led to some of our laboratories shift effort and work on coronavirus in a way that we didn't imagine we will do before. And the outcome of that is innovative discoveries by those that didn't think about the coronavirus in a way or the conventional way. You're going to hear about one of the laboratories, the Chanda Lab, who is going to take you to a tour outlining the research and the impact that came out from this type of work. The restrictions that imposed on us all due to the COVID infection were enforced here in the Institute, but nonetheless, we were able, and we are able nowadays, to work in distance with masks, and the laboratories are humming almost as they were before, dedicated people that are doing their utmost to address those unmet clinical needs in cancer. The complexity of COVID is now much better appreciated, given that we learn to recognize how this virus impacts not only the cells that it infects, but also other organs and systems in our body. The immune system being one, the coagulation system being another. And it's clear to us today that the solution is likely to encompass the use of combination drugs that will be needed to work together. And that knowledge comes from basic research work, clinical work, and models that we are using to identify the best possible combinations. COVID has impacted the cancer patients, and that is at least in two different fronts. The first is patients that have cancer and have been subject to therapy may not be able to continue on the same therapy, since those weakened their immune system, weakened systems that could have been exploited by COVID-19. So they had to change course of action, course of therapy. The second had to do with our ability to early detect cancer. Mammography, colonoscopy, just a couple of examples of analysis or early detection measures that are undertaken routinely help to identify patients at risk and treat them with preventive measures or with the proper measures in time to prevent cancer from developing and becoming more aggressive. And this has been delayed because most of our efforts in the last 6 or 8 months were devoted to treatment of COVID-19. Before I hand it over to Helen, who is a member of our Cancer Advisory Board, I would like to thank the Cancer Advisory Board for making this event possible, for their commitment and engagement in what we do here as researchers in the Institute, and Helen in particular for moderating this event today, hoping that you're going to enjoy the tour, virtual tour, that you're going to have over the next hour or so.
Thank you, Dr. Renai. It is a joy to work with you, and thank you for all the ways that you have been kind to me, even with some of my not so smart questions. My name is Helen Ekman, and I'm a member of the Sanford Burnham Community Advisory Board for the Cancer Center. And thank you for joining us. And we hope that next time, like Dr. Renai said, that we could meet in person and have you here. But we're delighted that you've come from around the world to join us today. I'm fortunate to be part of this Cancer Center Advisory Board for the last 20 years. And I was first diagnosed with cancer over 30 years ago. And this group has provided me with a way to make a difference with cancer research. And I have been able to lend my perspective to the scientists, and they actually are interested in hearing what I have to say. We've asked you to be a part of this today and to learn from us about what is going on, and these are amazing events. So I do want to say that I wouldn't be alive today if it wasn't for some of the research done at Cancer Research at this center. I am actually taking a compound of drugs that are keeping me alive, and they were first researched here. How grateful can I be? So cancer has likely touched almost every one of us that is listening to here and seeing us today, either yourself or someone that you love or know. And the pandemic makes this journey with cancer just more difficult. But it does not mean that we can be less vigilant with our screenings and treatment. And we find that some women are not getting their mammograms now, and they need to. We need to continue with that. If my cancer was first diagnosed with a mammogram, I wouldn't be sitting here without that diagnosis. So please continue to do that. So we've also learned through this pandemic that science matters. Science is important. And so today, the global pandemic has really made this more clear to us. So our mission is to find treatments and cures for cancer, and it's more important today than it was before. The work here done at Sanford Burnham Previs is critical work. Only if we continue fundamental and translational medical research will there be future treatments and cures. As a community, we must work together toward our goal of eradicating these devastating diseases. I speak for the entire Cancer Center Community Advisory Board when I say thank you for being with us today. And we hope that this open house will be illuminating. Today, you're going to hear from 3 labs, okay? And you're going to find out unique opportunities and research that they're doing. And you're going to actually be able to step virtually into their labs and see exactly what's going on. So following the tours, the 3 tours, we're going to have Q&A. So before we begin, we want to give a special thank you to the American Cancer Society today for joining us. And we also want to tell you that you're going to be hearing from Dr. Jenna Hope, who is one of their new fellows. So let's begin today now with the first tour, which is the Chanda Lab. And they have screened thousands of existing drugs to be able to identify those drugs that we already have access to and find out how they can be repurposed for the treatment of COVID-19. This is very exciting. So Dr. Chanda, take it away.
Thanks, Helen, and also thank you for your involvement with SBP. We really appreciate all your input and contributions. So my lab works, so prior to this year, we looked on a number of different viruses, including HIV, Ebola, influenza, Zika, understanding how they infect cells, how our immune system responds to those viruses, and developing novel curative approaches for those viruses. About late December of last year, I received an email from a postdoc of mine who was traveling home to China that would change the course of our lab, and the information contained in it would also change the course of everyone's life as we know it. He wrote that there was an outbreak of pneumonia in the Hubei province in Wuhan, China, with unexplained causative agent. And about a week later, he wrote back the words novel coronavirus. So when a virologist hears the word novel coronavirus or novel influenza virus, we immediately go into war footing. What we decided to do, because typically it takes between 10 to 15 years to bring a new drug to market. Of course, we didn't have that kind of time. We decided to look at a collection of 12,000 existing drugs that have been evaluated by the FDA to find drugs that we can immediately spring into action to treat coronavirus. Now, the person that I'm about to introduce, Laura Martin-Sancho, was one of the first people and probably the first person in San Diego to receive the virus from the CDC. She heroically sprung into action, grew it up, and we were able to conduct studies to find both new drugs and understand the virus. Prior to her travails in COVID for the last year, she had been working on influenza in the lab. And before I turn things over to her, I just want to acknowledge Laura and the other folks in the lab that have been really working themselves to the bone, putting themselves in danger, working with this virus, and leading a way towards eradicating this virus and getting us all past this pandemic. So with that, Laura, we look forward to both your tour and the talk that you're going to give.
Hello, I'm Laura. Thank you very much, both Helen and Sumit, for such a kind and nice introduction. So today, as I said, there will be two parts. So first, I'm going to give you a very small tour in the lab. Unfortunately, I wish I could give it to you in person and show you the benches and where we do the research that we do here in the Institute. But considering the ongoing pandemic, we thought this was the most safe option. So as I said, first I will play a video and then I will give you, I will show you some slides that show some of the results that our research came up with. So in the video, what you're going to see, as Sumit mentioned, we were one of the first people in San Diego and among probably across the US that got the virus and that had the honor to propagate it, infect cells, and we have been even sharing protocols with other laboratories. So what you're going to see is the moment actually that we received that virus. So it came in a very steel box. They were like different containment levels. So then you will see how somebody hands me that box and then I bring it to the BSL-3. But without further ado, please let me share my screen and I will be playing the video for you. So, so this is the moment. So this is me back in March of earlier this year. I feel like I look much younger than I look at the moment. So then she's basically explaining the virus had to be delivered in hand by somebody who actually had the credentials to work in a high containment laboratory. So she's basically telling me that inside of the box, there will be another box, and inside of that box, there will be another box, and then there will be a screw cap that then I could bring inside of a sterile cabinet and then open it there. And then again, there will be another bag and then finally the vial. So then that was the door of the BSL-3 facility before we enter. So this will be the door that actually leads to the actual BSL-3. BSL-3 stands by Biosafety Containment Level 3. So it is only highly trained people can actually access. So this is the ante room where actually I have to remove all my clothing. Then I put this this will be what hospitals are being used, these scraps. And then basically I have to have an external respirator. So you will see me now how I'm putting this cap over my head. This will be connected to these small charges that is around my waist. Basically, the idea is that I will not be able to respirate any inside of the BSL-3, that everything will be completely controlled. And on top of that, I'm going to put what is called HIVAC. So basically, no part of my body, there is no single centimeter of my body that will be in contact with inside of the BSL-3 that will be exposed. So then I have to make sure, so you can imagine, putting this on and off every time I want to do some work with the virus is actually pretty time consuming. So, but it's really important that we make sure that everything is sealed, that everything is properly zipped, so that there is no possibility of me getting infected or anybody else who will work in the BSL-3. It is very important, of course, we can see here the Tyvek. So I have the first pair of gloves. There will be a second pair of gloves that I will be putting in a minute. We have to seal very properly this first pair of gloves to the Tyvek because we don't want, as I say, any exposure happening. So it is very important to seal it very well. So I put it very tight. So then I make sure that there is no possibility. And again, this is the second pair of gloves. We use two different colors of pair of gloves. So the first one is blue and the one on top is black. So we are only allowed inside of the sterile cabinet when we work with the virus with the black gloves. And every time we touch something infectious and we want to get out of the cabinet, we need to replace that black pair of gloves. So this will be the BSL-3. Apologies for the video, maybe it is hard to see myself, but we only have a very small window. So this is the moment back in March when I opened the box. So as I say, inside of the box, there was another box and another box. And then there was this tube that we can see here in a plastic bag that actually this is the one that I can bring into the sterile cabinet. So the idea was that they will send us very few microliters of the virus and that we will have to propagate it to the extent that we can actually do a lot of experiments with them. So it was a pretty hard task. So now this is outside of the BSL-3. So this is the laboratory. This is how it looks. This is Sumit. You just met him a few minutes ago. This is our beloved Bunny. So Bunny is actually, it was helping us out with some of the experiments, and eventually only the infectious part has to be done in the BSL-3. But once we conduct experiments, and there are some follow-up parts that can be actually done out of the BSL-3. And I believe Bunny is actually pipetting a PCR. So this is a technique that we use pretty frequently in the laboratory, and eventually it is so we can detect whether there is a virus or there is no virus by doing some sort of amplification of the genetic material of the virus and then being able to run it in a gel. So as we can see, Bunny has very steady hands. So she is basically putting a master mix inside of the tubes, and then these tubes will be then subjected to different cycles of temperature that will allow, as I said, this amplification that is required for us to actually be able to visualize what is happening.
So these are the assay plates we use to query the drugs to see if they have antiviral activity. So each of these plates has 384 little slots. Each of those slots gets a different drug, gets some cells, and gets the virus, okay? For the drugs that don't do anything, the virus will go in and they'll kill those cells. For the drugs that block the virus, we'll see that the virus is no longer able to kill those cells. And so we zero in on those and then test them in additional assays to make sure they're actually blocking viral replication. So from this assay, we hope to get 30 to 40 different drugs, and then from there, we'll validate them in these additional assays. We hope to get down to maybe 3 or 4, which look promising, and move them into either animal studies or directly into human trials. This is how we measure how much virus we have. So these are what are called plaque assays, and these are essentially the virus killing the cells. And when you see bright purple, that means there's no killing. And then when you start seeing these little splotches, that's where the cells are dying. Okay, and so the more the cells are dying, the less of the blue stain these wells take up, there are more viruses in there. So that's how we tell how much virus we have and how much death the virus is causing to the cells. So imagine it as a funnel, and we start with 13,000, we get down to 50 or to 100, and then we get down to 2 or 3, and those 2 or 3 are validated in assays that are highly technical and can't be done in these very small formats.
So now that we have watched the video, I'm I'd like to share some of the experiments and some of the data that we have been generated since March, and I will be putting them, as I say, in context of the efforts of the lab in order to find drugs that could be used for treating COVID-19. So before we start into the finding antivirals and how does it work, I guess many of you have been listening to the news and been hearing about SARS-CoV-2 or the causative agent of COVID-19, about viral infections, and many of you will be wondering, but what is it, the virus? So then it is sometimes hard to find basic information that can help us understand what is coming from the news and what is actually going on. I think my favorite definition of what a virus is, it came from Jean and Peter Medawar in 1977. They say a virus is basically a piece of bad news wrapped up in protein, which is not very far from reality. Viruses have a very simple structure, in contrast to maybe other pathogens like bacteria, or in contrast to our cells. So basically they have three components. So they have the genetic information. So eventually this is the information that a virus needs in order to propagate, in order to replicate, to give their information to the progeny. So then all the proteins are being made. And this can be either DNA or RNA. And in case you think that these words, you probably have heard about them, and maybe as a refresher, DNA and RNA are actually the building blocks of our genome. So then if we think, for example, as a computer where every information is either made by ones or zeros, DNA or RNA is not very different from that, only that there are 4 letters, and then these 4 letters determine everything, how we are, for example, the color of our eyes and the color of our hair. So this is it. So this will be the first component. The second component is viruses have proteins. For example, everybody has probably heard about spike. So then the causative agent of SARS-CoV-2 has a protein, for example, that is called a spike, and this is required in order to get inside of our cells. So then there are proteins that recognize the receptors and then allow them to get inside of cells and infect. And then some viruses have an envelope that kind of goes around, and it kind of closes up and provides some spatial limitation. Some others have another different structure. So I think one of the most important characteristics of the virus is that they cannot replicate outside a host. So unlike, for example, bacteria who can actually replicate by themselves, viruses need to be inside of the host, either animal, human, or a plant, in order for them to replicate. And one of the reasons is that they are very small, so they don't carry a lot of information. They don't make a lot of proteins, so they are in general quite simple. So if we can look at the size of the virus, so this will be a human hair. If we amplify this, this will be maybe a particle of dust of pollen. If we amplify this, this will be relatively the size of a bacteria. A virus is even that, like it is really, really tiny. So again, it doesn't have the ability to replicate by themselves. So then they have to infect a cell in order to exploit all their proteins, so then they can replicate and form progeny. And another important thing is how the viruses transmit. There are many different routes. So one is through a vector, say for example, Zika or dengue viruses that, as Sumit mentioned, are viruses that we were already working in the laboratory. These transmit through arthropods or mosquitoes. The other viruses, like for example, HIV and Ebola, that they transmit through body fluids. And there are other viruses that are airborne. So this will be the case of SARS-CoV-2, that they transmit through particles and when people sneeze, or the saliva that we produce when we speak. This will be the way that viruses that are airborne will transmit. So the causative agent of COVID-19, the virus that we call SARS-CoV-2, it is airborne. And so then this is the way that the virus will then start infecting in the upper respiratory tract, and it will migrate to the lower respiratory tract. So the initial infection can actually end up with the virus actually damaging the alveoli, and this is why some people have some complications and have some problems to breathe and need a respirator. So this is an image that shows a very nice surface of cells with cilia and mucus, and here in red we will see the virus. But I don't want to get too many explanations or too much description about this because Sandra Leibel is actually going to be talking about that. So please stay tuned for her talk. So how can we treat these viral infections? In particularly, how can we treat the causative agent of COVID-19? I always say the best treatment is prevention. Actually, preventing from getting infected will then render treatment not needed. So I always recommend to wear face masks, to wash our hands very thoroughly. If you're sick, please stay at home. Try to limit all your social interaction. Hopefully the vaccine is it's coming very close, so then we can resume all these activities very soon. So usually there are two different ways of treating viruses. One is the vaccine. And again, this is our best bet at the moment for trying to stop the propagation of COVID-19. And the other one will be antivirals. Antivirals are very handy. I mean, they can either be used as prophylactic, so then they will prevent us from getting infected, or they can be used as treatment. So we are sick, we take an antiviral, and immediately we feel better. The caveat of this is that, and this is what Sumit was talking about, the novel design of antivirals can take from 10 to 17 years. So imagine when the pandemic of SARS-CoV-2 or COVID-19 started, we don't have 10 or 17 years to spare. So there is another possibility and there is another option, which is called repurposing. So what does it mean? So we do have cancer drugs, HIV-1 inhibitors, Ebola virus, anti-malaria drugs that are actually approved. And we know that these drugs are very safe, that people take them and they don't develop complications. So we know how actually our body will react to these drugs. So what if we could actually use these drugs and test them and see if they actually can prevent or inhibit the replication of SARS-CoV-2, the agent that causes COVID-19? So then that will mean that in months, we could actually have a drug that people can use. And this is exactly the case of remdesivir. This drug was designed to block the replication of Ebola, but we have seen that actually it can alleviate the symptoms and the replication of SARS-CoV-2. So how is this possible? If you remember from my previous or from my first screen, I said that viruses can replicate by themselves. They need to infect a human cell or an animal or a plant cell to replicate. So it is possible that some of these drugs that are targeting functions or are targeting part of the host that the virus needs to replicate, and then they could have broad spectrum activity. And that's why some cancer drugs or drugs that are designed for other viruses could actually be still working for treating COVID-19. So how does it work? How do we test? How do we find repurposed drugs? So this is again an image of myself in the BSL-3. So all these experiments were conducted in that facility. So how do we discover SARS-CoV-2 antivirals to treat COVID-19? As Sumit mentioned, we use a library of 12,000 compounds, and this was in collaboration with the University of Hong Kong. So how do you test 12,000 compounds? I mean, this looks like a really big number. So what we actually do, and Sumit was mentioning in the video, we have these sort of plates. They're as big as the palm of my hand probably, and each of them have 384 well pockets, and each pocket has a different drug. So suddenly 12,000 doesn't look so much when you can test 384 at a time. So we spot a different compound in each of these holes, then we add cells on top, and then we infect them with the causative agent of COVID-19, with the SARS-CoV-2 virus. So what happens, these cells are going to get infected, as you can see this one in red. And viruses actually generate what is called the cytopathic effect. The cell that the virus infects usually dies. And this is something that we can measure in the lab. We can measure if a cell dies or it doesn't die. And of course, if a compound in an inhibitor doesn't diminish the replication of SARS-CoV-2, we are going to see that the cells keep dying. But if we see that the cells are not dying, it could be that this inhibitor actually prevents the replication of the virus, and then we should flag it and follow it up for different assays. And different assays that we use in the lab include this. So this is N staining. So we have probes that are able to detect the virus. So this will be a monolayer. So this will be human cells. And then we can see that the cells, each blue spot will be one cell. And then we can see that these red staining marks cells that are actually infected. And this will be without drug treatment. But when we add remdesivir, we can see that this staining is almost completely gone, suggesting that this, and we knew it back in April, that this drug was working very good. And it is fascinating that eventually so many people are being treated and it's been so helpful. And I think Sumit mentioned that we expected finding around 30 compounds from these 12,000. He couldn't have been more right. I think that video was taken March or April. And actually now in November, when we actually finished the study, we did find around 30 promising antivirals that we are following up. And we are using different models. For example, we early on during the pandemic, it was clear that people that get infected from COVID-19 was losing the taste and smell. So we were wondering actually if the virus could actually infect the brain. So this was something pretty scary for us, and we wanted to test it in the laboratory. So in collaboration with the laboratory in UCSD, we actually, what they do is they are able to create or grow mini brains in small dishes. So then I brought them inside of the BSL-3, and to our surprise, so then this will be again human cells, as marked with these blue dots. And what we saw is that the virus can infect brain cells. But very luckily, we tested the different compounds, and we found these compounds, so fusbivir, which is an FDA-approved drug, that actually could inhibit replication of this virus in the brain. So it's actually very good news, and we are really happy and very honored that we actually could conduct these studies. So to finalize my talk, I would like to acknowledge some of the people that have been day by day helping us and trying by all means that we could work in a very safe environment and that we could be doing all this testing. So of course, I would like to thank my PI for the opportunity to work at his laboratory. And I have to say, all this work was conducted by Laura Riva, a fantastic postdoc, and Jin Shin, as well as Courtney Nguyen, who have done an amazing job. Again, I would like to thank Biosafety, and then the University of Hong Kong, University of California, San Diego, Mount Sinai, and of course the NIH for all the funding so that we could do these studies. So thank you very much for listening. And of course, I'm very happy to take any question that you may have at the end of the session. Thank you.
Laura, that was exciting and wonderful. Thank you so much. And thank you, Dr. Chanda, as well. This was just a wonderful, Dr. Laura Martin-Sancho, wonderful presentation. And thank you for all of the information. Young girls all over the world now are going to sign up to be scientists. So you guys have been on the front line of this, and I'm really pleased. So I would now like to introduce our next lab number 2. And this is going to be with Dr. Sandra Leibel and Dr. Evan Snyder, and they will be discussing creating mini lungs. I know I can't believe it, mini lungs for testing COVID-19 drugs. So take it away, we can't wait.
Hi, everyone. Well, I wish we were doing this in person. And I guess if we do our job the right way, next year, we will be able to do this in person. Hopefully, Sandra is going to share a video off of her screen that will be a little tour of our lab. This is me pondering the possibilities of stem cells. And I guess you should know that a stem cell biologist lives in a tissue culture room. So what are stem cells? I guess the best way to think about stem cells are that they are the seeds of the body or the seeds of a particular organ that you're interested in. I'm a neuroscientist, so we're very interested in the brain. Sandra is a lung biologist, so she's very interested in the lung. And in fact, I once had the privilege of giving a one-on-one lecture to Stephen Hawking on stem cells, And for him, I called it the Big Bang of the body. Anyway, most people think about stem cells as what you do and you transplant into a brain. And here I am looking at cells derived from stem cells, brain cells, and how they've developed. We also use them, however, to model diseases in a dish, mini brains, mini lungs, and use them to figure out diseases and to also figure out drugs against those diseases if we understand the targets and things of that sort. What we're looking at here is Sandra. Sandra and I are both neonatologists. We study the brain, we study the lung, and Sandra and I joined forces a number of years ago to figure out whether we could study certain baby lung diseases. In fact, we arguably have the only lung stem cell lab in all of San Diego. But when COVID-19 hit, we realized that we also had the opportunity to study this disease and to test the drugs that Sumit and Laura had figured out. And could we fast track those drugs into patients, particularly if they were FDA approved? And Sandra and I, but pretty much led by Sandra, so I'm going to turn this over to Sandra, created a massive lung team to start screening drugs, but also figuring out what cell types really was the virus hitting and what drugs would stop what part of that disease that we could fast track to patients. So with that, I'm going to turn this over to Sandra to finish the tour and also to introduce members of the team.
Thank you, Evan. Hello to everyone. I will continue the tour, the virtual tour of the Snyder Lab. So Dr. Snyder has kindly allowed me to do my stem cell derived lung organoid work in his lab. Here is Rachel McVicker, who's the graduate student that is working under me. And she is currently making little stem cell derived lung organoids, mainly ones that replicate the types of cells we see in the trachea, as well as in the nose. And which we all know, those are the ones that actually get infected first. So the way that we can study what types of cells we actually make from the stem cells is to do something called flow cytometry. And so that's what you're seeing right here. It's this big machine that detects little colors on the proteins of various cells. So if we're trying to make a cell that's called a cilia cell or has little hairs on it in order to move mucus around, there's also mucus secreting cells. Those two cells have a different kind of protein on them. And if we add an antibody to make the ciliated cell green and the mucus secreting cell red, then when we do the flow cytometry, then those will be separated by the machine and tell us, oh, look, you have 25% red ones, which are the mucus ones, and maybe 3% of the cilia, which are the green ones. So that's one way that we analyze the type of lung cells that we make. Another way is to actually use a microscope. So this is a fluorescent microscope that's being pictured right now in the video. And that we can actually use to look at live cells. So we don't have to kill the cells in order to examine what type of cells that we're making. We can actually just leave them in their little dish with their media that has all the nutrients in it for them to be happy and live, and actually be able to study how fast they're growing, what they look like, do they look a little sick if we're adding a different compound to study inflammation. We can actually visually, with this screen, determine the health of the cells and how well they're growing. So my talk is mainly going to be about the respiratory system or the lungs. As Evan mentioned, I'm a neonatologist, just like Evan, a clinician scientist. And my interest in the lung occurred when I was taking care of babies that were born with a mutation in the surfactant system, which I will explain what that is. But in general, it's a molecule, a soapy molecule that actually keeps all of our lungs open. So we are all currently here today and alive because of surfactant. So if a baby is born with a mutation, they are actually unable to transition from the fetal life to an air-breathing newborn life and need to be on the ventilator until they're able to get a lung transplant. And so I would get to know these families for being in the NICU for 3 months, and it really drove me to try and discover a cure. The problem with the lung field, the world of lung understanding and research, is that we actually don't have a good model system to study human lungs. We have animal-derived projects that we can examine and look at, but unfortunately, human lungs are very difficult to obtain. They're very difficult to grow in a dish. And so when a lot of previous investigators had figured out a way to add some compounds into stem cells and push them towards the lung, kind of mimicking development like a fetus goes through, that's when in around 2012, I jumped in on that bandwagon and devised my own differentiation protocol to make mini lungs in a dish. So what does this have to do with COVID-19 and cancer? Well, when I was recruited to UCSD, I was initially just interested in studying the surfactant system and lung development, but then I met a collaborator who was interested in infecting the lung organoids with respiratory syncytial virus, or RSV. And this is a virus that if a preterm infant gets when they're home, then they could get really sick with it. In us, we maybe get a runny nose, a cough, some sneezing, but preterm infants, when they get RSV, then they can actually end up in the hospital. And so we were trying to figure out why they are more susceptible to this disease. So both of us started working together to infect my little lung organoids with RSV, and then the pandemic hit. And at that time, we realized that Evan and I realized we were the only ones at UCSD, as well as probably all of San Diego, that actually had a human-derived lung model. And therefore, I quickly got BSL-3 certified, just like Dr. Martin-Sancho explained, wearing the full astronaut gear is what I call it, in order to be able to access the lab at UCSD and infect the lung organoids with the coronavirus, or the SARS-CoV-2 virus. So let me just walk you through this picture of how this virus causes lung disease. So here we have a whole lung showing the trachea, that's in white, and then the kind of meaty part of the lung that's in pink, and that's the more distal airway, and the trachea is the proximal. The virus initially infects the proximal lung cells, which include the ciliated ones, or the ones with the little hairs that Dr. Martin-Sancho showed a nice little picture of, as well as the mucus secreting cells. And then in some patients, they have no symptoms. So they're called asymptomatic carriers. And therefore, even though they got infected, they don't know it, which is great for them, but not great for others that may be around them and may be unknowingly infected, which is why masks are the way to go. For others that aren't so lucky, they get infected in their upper airway, and the virus travels all the way down to the lower airway, which is depicted in this picture here. And you see these kind of grape-like structures. These are called alveoli. Now, alveoli are made up of two cell types. If you look at this picture up here, you see these flat pink cells. These are alveolar type 1 cells. They're really flat, so then we can exchange oxygen and carbon dioxide across from them with this capillary or blood vessel that surrounds the alveolus. These white, larger, kind of dome-like cells are called alveolar type 2 cells, and these are the ones that actually secrete surfactant, which allows the lung to remain open even when we're breathing out. So both of these are extremely important, and most of our lung is made up of these type 1 and type 2 cells in order to help us breathe, exchange oxygen and carbon dioxide, and maintain the lungs in an open state. So what happens when SARS-CoV-2, or the virus causing COVID-19, enters these cells? In some patients, they have an intact immune system, and Dr. Jenna Hope will definitely speak more about that. And they're able to clear the virus without any damage to their own lung cells. But in those that are not so lucky, or those that have comorbidities, such as COPD, or lung cancer, or hypertension, congestive heart failure, there is something about not only the structure of their lungs, but also their immune system that kind of overactivates it, in the sense where the lung cells recruit or bring in the immune system to help them get rid of the virus, which you can see happening in this picture right here, where the virus starts infecting the cells and they start dying, which creates holes in these alveoli, which allows fluid to enter. And so they start calling for these immune cells, which you see here are macrophages and various other ones, to come in, fight the virus off, and hopefully heal the lung back, which some patients it does happen. In others, unfortunately, this lung damage progresses into the severe form. And in the severe form, the immune cells that are recruited, they not only try and clear the virus, but all of the proteins and cytokines that they're secreting in order to kill the virus is unfortunately also killing the body's own cells. And that's what we see in COVID-19 in those patients that end up on ventilators. And so this is what we're trying to understand with our model system. So modeling disease and drug discovery, as I've mentioned before, the mouse model has been indispensable to understanding how our lungs function, how the immune system functions in that setting, as well as with all the different blood vessels, all in combination. But unfortunately, the mouse is not a human being. And we know that even in their lung differentiation, they completely take a very different path. And so it's really important if we're gonna study drugs and see how they affect human lungs, that we actually use a human model in a dish. And so with the discovery of induced pluripotent stem cells back in 2007, this whole field of regenerative medicine and making little organoids was really set. And so this is just a picture showing how we make these stem cells, which then we can go forward and make the organoids. So you could see here, I can take any of you and take a little bit of your skin, grow out your skin in a dish, and then I can add what are called some reprogramming factors or Yamanaka factors for the scientists who discovered these. And then I can change your skin cells. We can also use hair cells or white blood cells and actually change them into what are called induced pluripotent stem cells. All that means is they can make any organ system in our body. Pluripotency, that's what that means. And so I can take these pluripotent stem cells and let's go straight into the reprogramming part of this diagram and show you that we can make what used to be your skin, and now we're suddenly making the alveolar type 2 cells, which I just said were very important in making the surfactant, the upper airway cells, such as the ciliated and the mucus secreting ones, and even blood vessels and immune cells. And we can take all those cells and perform high throughput drug screening and actually look to see which drugs work for that specific patient. So it's not a one size fit all like we do right now in clinical medicine, where we think, okay, well, I guess we only have this one medication, so hopefully everyone's going to respond to it the same way, which we know, in fact, is not true. We all have our own genetics. We all have our own epigenetics. And so therefore, we're going to react to medications in different ways. And so if we can take patient-specific iPSCs, make them into specific organs, and the brain is included as well, then we can actually test various medications and see not only which ones are going to work, but which ones should be avoided because maybe they have bad side effects. And another way to utilize all these organs, mini organs in a dish, is transplantation. So this is great because I told you that these little babies that I was taking care of, they were waiting for a lung transplant in order to be able to go home. But if I can take their skin and make little lungs and then retransplant them into their bodies, number one, it's going to be their own cells. So they are not going to reject it. They don't have to be on any immunocompromising medications in order to suppress their immune system. So this is definitely the future of regenerative medicine and making all these various iPSC-derived lung, any type of organoids. So what about our own lungs in a dish? So this is just showing you how the differentiation process actually works. So this is the stem cell, and then it goes through one of the germ layers in our bodies, which is called endoderm. And then we plate them down into a 3-dimensional matrix, and they take about 3 weeks to grow. And what you can see here is what they look like for those 3 weeks as they develop into mature mini lungs in a dish. So you could see they start branching just like mini lungs do. And then here we add a specific cocktail to mature them. And you could see that they start outpooching these kind of balloon-like or grape-like clusters that we saw before. And then the mature ones end up being these nice, large, clear, circular lung organoids. What we can also do, and we're currently exploring right now, is trying not only to make lung organoids, but to make lung tissue organoids. Because as I had shown you in the diagram, it's not just simply having a lung cell that's important. You also need a component of the blood vessels, as well as a component of the immune system. And if you have all those components, then we can truly represent human lung tissue in a dish in order to be even better with our high throughput drug screening, or eventually the hope of transplantation. So here we have endothelial, which are blood vessel cells that are in green inside our lung organoid. And here we can also add macrophages, which are the immune cells that enter the lung in order to help against infection. They're seen here in red. So this is something that we are also making out of the iPSCs. Therefore, I can have one patient and make all of these different cell types in order to truly mimic their own body chemistry and genetics. So this is what the lung organoids look like after being exposed to that specific maturation cocktail. And so you could see that they go from being branching, dense, and small, and really growing into those larger grape-like bubbly structures. And so what we've also done in parallel is I work closely with a cardiothoracic surgeon, and she does lobectomies on adult patients once a week. And so she calls me as soon as the lung tissue leaves the OR and goes to pathology to get examined. And I go to the pathology lab, I collect a piece of the lung tissue that they're not going to need in order to evaluate what's wrong with it. And then I'm able to actually grow it in multiple ways. So I can do it what's called an air-liquid interface. And that just means really focusing on the upper airway cells in the trachea. Or I can do lung organoids out of the human tissue. And these definitely look a lot like my stem cell derived lung organoids. And lastly, what's really cool is we can inflate the pieces of lung tissue and then slice them up into tiny, tiny thin slices. And these are called precision cut human lung slices. So this is the lung that's preserved. So it's got the immune cells, it's got the lung cells, and it's got the blood vessels. And we can keep those pieces alive in a dish. And we've been infecting those with the SARS-CoV-2 virus as well. And so that's the virus infecting the cells. I don't have any cool videos like Dr. Martin-Sancho has about being in the BSL-3, but as you guys have seen, it's a very intense procedure and process to get in and out. But thankfully, you know, what we've gotten out of it has been completely worth it. And both of the human-derived lung tissue, as well as our stem cell-derived lung organoids, are infected. And we have confirmed infection with SARS-CoV-2. And we've been working with a lot of collaborators at SBP as well as UCSD to test various compounds to see which ones can block the viral entry and reduce infectivity. And so in summary, using our lung organoids to study COVID disease, you could use the patient-specific lines. Therefore, we can not only look at just making the organoids themselves, but we can actually choose iPSC lines based on gender, various ethnicities, and actually try and mimic what we're seeing in society when it comes to the disparities that this disease causes. We can also work with other collaborators that have discovered various genes that may be important in this virus spreading and knock them out and actually study to see, does this actually help? We can also expose them to nicotine and mimic lungs of smokers that are definitely at highest risk of getting these infections. And then finally, as I've said before, use drug screening to avoid the animal model. Animal models are great, but they really don't mimic 100% of the time what truly happens in the human body. So it's very helpful to directly test the human cells for effect. And finally, just to leave you on the hope of future cancer studies, we're also currently growing lung cancer organoids. So usually lung cancer has always been studied in a dish, but just kind of flat cells that, you know, we've been able to test different compounds on. And then the lung organoid world has really seen that they have to be in this three-dimensional way in order to truly replicate what's happening in the human body. And so we've started working with many lung cancer scientists at SBP to grow their lines in a three-dimensional way, and then try and perform radiation, radiotherapy, chemotherapy to determine which ones can actually help those patients with these various lung cancers. And also I'm taking these cells into the BSL-3 with me and infecting them with the SARS-CoV-2 virus in order to determine what is it about patients with lung cancer that puts them at higher risk of getting this disease. So thank you for your time. And I just want to thank again, Dr. Snyder, for allowing me to work and support me. We're on a grant together to study the lung tissue organoids after being infected with SARS-CoV-2. And then everybody in all of the various teams in the lab working on both brain and lung organoids, and all the collaborators and mentors that I've been lucky to have at SBP and UCSD.
Well, thank you very much, Dr. Sandra Leibel. That was amazing. Thank you, Dr. Snyder, for sharing this information with us. And we're learning about growing these lung organoids and from stem cells. This is wonderful research, and thank you for sharing it with us. So now we're going into our third and final lab, and we're going to be meeting with, it's called the Bradley Lab, and they're going to be discussing cancer, COVID-19, and our immune system. And I just have to say that Dr. Linda Bradley is one of my heroes, so take it away.
All right, thank you so much, Helen. And
You're welcome.
All right, so I'm on, I know that everyone can hear me. I want to let you all know that, like Helen, I'm a breast cancer survivor, and my husband is a colon cancer survivor. And as such, I can empathize with many of you in the audience who have experienced cancer and cancer treatment, and either as a patient or as a family member or friend of a patient. So I'm an immunologist, and my research focus over many years has been the cells of the immune system that destroy cells that become infected with microbes such as viruses, but also destroy cancer cells. These cells are known as cytotoxic T cells because of their killing activity. And we study these in the context of both virus infections and cancers, particularly in the settings where the immune response becomes ineffective. Under these conditions, T cells continuously are engaged in immune responses that ultimately fail because of exhaustion. They're functionally impaired and can no longer be effective. So in our studies, I should say these is from studies of viruses that originally led to the recognition that T cell exhaustion occurs in cancer, and of course that immunotherapy is one of the ways to reverse this dysfunctional state. However, that was back in 2013, the breakthrough of immunotherapy in cancer. And here we are now in 2020, and we know that many patients do not respond to these treatments. And in my lab, we are studying how the T cells become exhausted and how to prevent it. And also, what are the attributes of the T cells where their function can be improved. In these studies, we've also identified a new inhibitor of T cells in chronic virus infections that can be targeted in tumors that are resistant to the current treatment with anti-PD-1 therapy. Now, with the emergence of the new pandemic, coronaviruses that causes COVID-19, as you've heard, we are continuing to learn about the range of immune responses that can occur to a new virus infection in the population for which there is no immunity, and what needs to be done to achieve protection. We are also discovering how this new coronavirus can disproportionately impact vulnerable populations, and these can include cancer patients whose immune systems are weakened by various treatments. So I'm going to now introduce my American Cancer Society Senior Fellow, Dr. Jennifer Hope, who came to my lab with expertise in training, looking at T cell responses to virus infections that included influenza and HIV, as well as cancers that included lung cancer and melanoma. And she is now continuing her work on reversing metabolic limitations on effective T cell responses to cancers. So now I'm going to turn it over to Jenna. Thank you.
Thanks, Linda. So we recorded a video showing everyone a tour of the Bradley Lab and really tried to include all the members of the lab. So we're going to share that with you now.
Hi everyone, thank you for joining us. I'm Jennifer Hope. I'm a postdoc in Linda Bradley's lab here at Sanford Burnham Prebys, and we're really happy to show you guys around our lab and what we do in a regular day at the lab. So first we're going to go down here. This is Hannah. Hannah is a research technician in the Bradley lab. And Hannah, can you tell us a bit about what you're doing?
Yeah, so right now I'm working on screening cells for flow cytometry. These are cells from our mouse line, so we have to screen all of our mice to make sure that they're either expressing the specific genes we want them to, or not expressing certain genes that we don't want them to. And then these will get stained with antibodies and then run on the flow cytometer to check what's on the surface of these cells. So these are all the different antibodies that we use to stain. So different fluorochromes are different colors, and then they're bound to antibodies that will bind to certain things on the cells. So for example, if we want to look and see if it's a CD8 T cell, we can bind that and then that'll show up on flow cytometry. So right now I'm mixing up the staining cocktail that we're going to use to stain these cells. And these different antibodies will bind to different things on the surface of these cells. So I dilute them in solution, and then we'll mix it up with all the different antibodies that we want to use, and then put it on the cells, and then that will stain them so that we can look at what's on their surfaces later. So flow cytometry staining is only half of the way that we check our mice. We also use PCRs to screen them to check and see if they have the specific genes that we're interested in or not. So over here is where we do that. This is the area that we use to mix up the PCR reactions. And you may have heard of PCRs before. It's very similar to what they're using to screen for COVID, but we're using it to check for specific genes within our mice. So we mix up the PCR reactions over here, and then we put them in our thermocyclers for them to run. And then to get our readout, we'll take them over here and run them on a gel so we can see the specific bands that are made from the PCRs to see if they have the genes or not.
So now we're going to switch gears to our next research technician, Ashley. She's working on some of our tumor cell lines. Hey Ashley. Hi. Do you mind telling us a little bit about what you're working on?
I'm doing tissue culture work right now with our tumor cell lines. We work with different types of tumor cell lines here. We work with mesothelioma and different types of melanoma, which is a skin cancer. And right now I'm going to be replacing the media in them right now to give them more nutrients and to make sure that they grow well.
So now we're in the flow cytometry core facility. So this is where everybody at the Institute can come to run flow cytometry samples. Here is where we have our sorting facility, where you can see we have 2 sorting machines in biocontainment units. And this is where we can actually do our biohazardous sorting if we need to, or regular sorting with human samples. And over here are the analyzers. And so there's 2 kinds, but they basically do the same thing. The one that we use most often is this machine called the X20. So Una from our lab, she's currently running some flow cytometry samples. And Una, could you tell us a little bit about what samples you're running right now?
Hi, I'm analyzing the transcriptional factor expression levels in activated CD8 T cells. So if you can see, I already labeled these cells with fluorescent labeled antibodies. So there is a CD8 T cell population, and I'm looking this transcription factor with FITC labeled cells. So we can compare the expression levels using this intensity of fluorescence.
This is our main tissue culture room, and it's also where we do the majority of our work. And she's using something called CRISPR-Cas9 right now in order to target a specific gene for deletion. But because what she's doing is so important, we're going to let her focus on what she's actually adding to the cells, and I'm just going to talk us through the process a little bit. So right now, she's adding some complexes to her cells. She has primary T cells, meaning that they came straight from a mouse rather than a cell line. So in that way, they're very similar to what we would take out of ourselves, our own bodies. These cells were activated or non-activated so that we can look at different types of conditions. And now, by targeting this specific gene, she can then use something called an in vitro T-cell exhaustion assay, which is develop something that we see in cancer patients called T-cell exhaustion. Essentially, our T-cells become so tired from fighting against the cancer cells that they become functionally exhausted and they lose a lot of energy and ability to kill off those tumor cells. So instead, by using this in vitro assay, she can evaluate if this gene deletion enhances that T cell function and makes them better able to fight off and kill cancer cells. Now we're going to visit Dennis Otero. He's a staff scientist in the Rady Lab and an expert at T cell signaling. Right now he's working on some histology images, so let's see what he's working on. Hey Dennis.
Hello.
Do you mind sharing with us what you're working on?
Not at all. So what I'm looking at here are sections of tumors from a tumor model in mice that we use to study the immune response to cancer. We use a similar antibodies that we do for flow cytometry. These are fluorescently labeled, in which case we have a fluorescent microscope in order to identify those fluorescently labeled cells. Now, what I have here up on the monitor are some pictures I took of this tumor cell showing different immune cell types which can be present within the tumors. In red, we have helper T cells, which, as their name suggests, they promote the immune response. And in green, we have cytotoxic T cells. These are the cells that actually kill the cancer cells. Now, one important thing to know when we're studying these different cancer models in the mice is whether or not the immune cells can actually get into the tumor. And that's what I'm identifying here. And now that we know that the T cells can go into the tumor and these cytotoxic T cells can attack the cancer cells, we can start studying the immune response and whether or not they're amenable to to inhibitor receptor therapy.
So this is where the lab officially begins with Una's spot and Dennis's spot. We each have individual lab benches where we can work at, as well as the shared space where we do all these different techniques. Down this bay is where I sit. It's also where we have some of our analysis computers. For example, we have this PC here where we can look at the metabolism of the T cells. In this particular case, this is a readout of T cell metabolism using a method called the seahorse. This is also where we do a lot of our flow cytometry staining in general. So we have different diagrams such as shown here, which show all the different types of colors that we can use in association with our antibodies. But at the end of the actual experiment, now we have to do some analysis. And that requires using a specific software on our computers. So I'll show you what that looks like. A lot of people today have talked about using antibodies and using flow cytometry or fluorescence to look at a readout of how our immune cells are actually functioning. So I'm going to show you what it actually looks like to analyze flow cytometry data. In this case, this was an experiment similar to what Una was doing with an in vitro T cell exhaustion assay, where we can look at how the cells actually respond to different therapies. In this case, we're looking at something called cytokines. Cytokines are like the bullets of the immune system, our T cells in particular. These are what help to exert a specific function and kill cancer cells. And what these flow plots here show, where every single individual dot is a unique T cell. And we can see in this condition, we have a very nice population in this quadrant in the upper right corner. This is looking at the cytokine interferon gamma as well as TNF alpha. You may have heard about TNF alpha in context of different therapies. And what you can see is that in the second condition, we have a lot less cytokine production. And so this is a way that we can tell if our T cells are responding or not to our different treatments. Another technique that we commonly use is different types of sequencing. We can do bulk RNA sequencing or even down to the single cell level, looking at what's called the transcriptome of an individual cell. And with the Institute, we have access to a particular software that allows us to take that data that we've generated from sequencing and apply it to what is known in general about T cells and immune cells to see if there's anything in particular of interest that's been modified in response to our treatment. So as an example, one of the things that we can do is bring up what's called a pathway. And this is what is generally known about in the field about how particular molecules are regulated in response to different stimuli. And we can see that based on our particular data, certain molecules, in this case CD3, which is expressed on T cells, is differentially expressed upon our treatment. And this is just one more way that we can look at how our particular therapies are modifying our T cells to make them better in fighting against cancer cells. Thanks again for joining us. We're really excited to have you come here and visit us actually in the lab after COVID is over, where we would be very excited to share with you more about what we do on a daily basis, trying to find out how to re-energize our T cells to fight off cancer cells. Thanks.
It really is our pleasure to share with you what is going on with the immune system. So today I'm going to be talking about the immune system in general and what this means in respect to cancer. So there's a lot of discussion going on about what immunity really means. And so we thought it would be important to start off with a general description about immunity in general. So let's begin by talking about what the immune system is. It's our built-in system of defense against foreign pathogens. So what does this mean? Well, our immune system is highly specialized. It's designed to identify if a cell or a protein that's not in our body originally came from there. Basically, if it's self, or if it's not meant to be there, which we call non-self. The immune system itself is made up of many different cells, all derived from the same precursor, the hematopoietic stem cell, like what Laura was just talking about with stem cells. But this cell resides in our bone marrow. And ultimately, from that one cell, it can develop into all of the many different cells shown here, each with a highly specialized function that assists our body in clearing pathogens, infections, or cancer cells that have developed. Besides our red blood cells and platelets, all of these other cells shown here are called white blood cells. And we often think about our immune system as consisting of two main branches, the innate immune system and the adaptive immune system. Now we need both arms of the immune system to work together in order to get the most beneficial and the strongest response against pathogens. But today we're going to be focusing on the adaptive immune system because these are the cells that respond to vaccines. The adaptive immune system is activated when specialized cells called antigen-presenting cells essentially sound the alarm and bring foreign pathogens to T and B cells. Now, our T and B cells are in constant circulation throughout the body, traveling along the lymphatic system. And along this route are lymph nodes, which essentially serve as the rest stops for our immune cells on this lymphatic highway as they patrol for foreign invaders. Now, in the case of a viral infection like coronavirus, these adaptive immune cells will recognize unique proteins that are associated either with the virus or the virally infected cell. So in this first video here, we see that our B cells and our T cells are rapidly moving within the lymph nodes. And then in the second video, we have antigen presentation by our dendritic cells to T cells, shown here as the red and the green cells. Now, this is all very important because once these cells become activated, they will expand to form millions of these highly activated cells that actively seek out and destroy the infected cells that we see in our bodies, such as those infected with coronavirus. So let's take a closer look at what happens when our bodies are exposed to coronavirus or any new viral infection. When we're exposed to a virus for the first time, our adaptive immune system is a bit sluggish in responding. While our innate immune system tries its best to contain the virus, often we see the viral infections spread within the body shown here in red. Within a few days, however, our B cells will kick in and release a first wave of antibodies that target the virus. And then soon thereafter, our T cells become activated. They expand and seek and kill the infected cells. Then a second wave of B cell responses, even more specific than the first, will start, and together these adaptive immune responses allow our body to take control and stop the viral infection. After the viral infection is cleared, we're left with a smaller pool of virus-specific antibodies and virus-specific T cells, which remain in our bodies for a very long time. Now, these are called memory cells because they remember that our bodies saw a particular virus before. Then if we're exposed a second time to the same virus, our memory T and B cells kick in much faster with more specific antibodies and stronger T cells that help us control the viral infection faster and limit how long we actually feel sick. This system of generating protective levels of immune memory is the major principle behind vaccines and building up what's called herd immunity, which you may have heard about in the media. Herd immunity is when at least 70% of the general population has a protective level of immunity against a given virus. When we receive a vaccine, we're essentially tricking our bodies into thinking that we have an active infection, even when there is no active virus present. But our immune system responds as if there is, which is why sometimes we may feel sick after getting a vaccine. But importantly, our B cells will initiate that first wave of antibodies, followed quickly by T cell responses and the second, more specific B cell antibodies, again forming that vaccine-specific adaptive immune response, which eventually form the virus-specific memory population in the same way as if our bodies saw a real virus infection. But importantly, we didn't have to go through everything that comes with an actual infection. We don't have to risk exposure to the many side effects that accompany a severe respiratory virus infection like coronavirus. But if we do come across and get exposed to the virus later, our body is prepared. It's ready to quickly respond and quickly bring that viral infection under control with decreased viral loads and helping us feel better faster. Now, many companies around the world are actively working towards the development of a SARS-CoV-2 coronavirus vaccine, and they're exploring many different approaches towards that ultimate goal of wide-scale protection against infection. Now, in this figure, each dot represents a unique vaccine design and in which stage these vaccines are in development. So why do we need to have all of these different vaccine designs? Well, because each of these have a different type of immune response that's generated, and we don't know yet which one will be the most effective in wide-scale immunity against coronavirus. Now, as of the end of October, we had over 50 vaccines in early stage or different stages of clinical trials, all moving towards hopefully limited approval or full approval. And you may have heard about the recent press release from Pfizer talking about their vaccine efficacy, showing good early signs of generating protection. These next few months are going to be very exciting as we learn more from each of the different companies about how well each vaccine candidate is generating protective immunity. And research studying how the immune system responds to SARS-CoV-2 virus infection are ongoing active areas of research. And every day we're learning more about what defines a good immune response to COVID-19. For example, one thing that we know about COVID-19 patients is that some of them will experience severe lymphopenia, meaning that there's a sudden decrease in their T and B cells. Since these are the cells that are going to drive that effective immune response, that could be a problem in clearing the infection. Now, although all of our cells in the immune system play important roles in fighting off an infection like SARS-CoV-2, there's one cell type in particular that actively seeks and destroys virally infected cells. And as Linda mentioned, these are our cytotoxic CD8 T cells. And actually, these are the same exact kind of cells that are responsible for recognizing and killing cancer cells. Now, this is perhaps one of my favorite videos to share. It's not from us, but I love how you can clearly see how our CD8 T cells are serial killers, going from one tumor cell to another and inducing tumor cell death. In this video, the round cells are the CD8 T cells, while these long cells are the cancer cells. Pay close attention as the cancer cell turns red, because this is the exact moment that the CD8 T cell has initiated the death and destruction of the cancer cell, and you can see it collapsing in and of itself. Then it moves on and kills the next one. So as you can see from this video, our CD8 T cells are absolutely essential to helping us fight off and beat cancer. Our immune system is normally finely balanced, but when facing cancer, the cancer cells can outcompete our T cells, leading to cancer cell growth. Now, Linda mentioned cancer immunotherapies, and these are crucial because what they do is help to promote our immune system and specifically increase our T cell responses to cancer. What this does is help tip the scales in our favor, increasing that T cell killing and limiting the spread of cancer. In the Bradley Lab, we've discovered a new immunotherapy target that helps our T cells keep tumor growth in check. When our immune system is fighting against cancer, our T cells are leading the battle, moving from cancer cell to cancer cell, just like in that video that we watched. However, in this process, it's extremely energy consuming. Our T cells become exhausted because it's like climbing up of a mountain with a heavy backpack that keeps getting heavier with every step that you take. But when we found is that with our molecule, we're essentially giving our T cells that extra hand to get them over the top of the mountain. And what we see on the right-hand side here is real life data from our lab showing that when we target our specific molecule, this results in decreased tumor growth. Basically, we can better control the growth of these tumors compared to just regular normal cells. But finally, I just want to briefly touch on the impacts of COVID-19 disease and cancer, because while all of us are adjusting to new normals, cancer patients in particular are severely affected by this, trying to adjust their treatment regimens and schedules to avoid risk of infection. We know that a weakened immune system is a side effect of many different cancer therapies, and this puts patients at greater risk of infection with SARS-CoV-2 and the development of COVID-19. We also know that there are certain types of cancers that are associated with increased COVID-19 disease severity. Specifically, those individuals with lung cancers, like Sandra was speaking about, or blood-based cancers, are particularly at greater risk for severe disease after exposure to SARS-CoV-2. And so we know that although all patient responses are different, we each can do our part to keep everyone protected and as safe as possible as we move forward in this new normal and trying to figure out what the long-term impact is of this disease and cancer. So with that, I would like to thank you very much for your attention and thank all the members of the Bradley Lab, as well as SPP core facilities, our funding providers, the NCI and the NIH, and of course the American Cancer Society. If you have any questions, happy to take any emails. My email is here, jhope@sppdiscovery.org, or you can use this QR code to visit the Bradley Lab and check out the other cool research that we're doing. Thank you.
That was wonderful, and we love those T cells, those cancer-fighting cells. Let's go T cells. Thank you very much, Dr. Hope and Dr. Bradley, for sharing what you're doing in your work. We're now going to move into the question section, and if you want to put a question, go down to the bottom of your screen, look to the Q&A area, and if you could please put your questions in there. We're going to try to get to them, and if we don't get to them today, we'll try to get back to you with them. So, thanks very much for the tours. So how we're going to handle the Q&A is that I'm going to read the question, and then Dr. Ronai, who was the first person who opened up this seminar today, he's going to say who's going to answer the question. So we're all ready to go for question and answer. Okay, let me see our first question. Okay, if someone gets COVID and recovers, will they be immune to the virus? Okay, Dr. Ronai, who would you like to have answer that question?
Let's start with Sumit.
Okay, thanks, Zev. So that's a great question. And there's no easy answer to it. So what it looks like is that if you get mild COVID, you generally don't develop immunity. If you get severe disease, if you have to go to the hospital, you will build up some level of immunity. I think the key question is going to be, how long does that immunity last? This is going to have implications for both people who've had COVID as well as the vaccine. Most coronaviruses, we somehow learn to forget real quickly, which is why we get the common cold every year caused by coronaviruses. Hopefully this will be different, but the jury's still out on the durability of the immune memory for SARS-CoV-2.
Thanks.
Thank you, Dr. Chanda. So our second question is it true that patients with severe COVID are not being put on respirators as much as they were during the beginning of the pandemic? Is this true? Why are we not? Dr. Renai, who would you like to have answer that?
You want to have a stab at that, Sumit?
Yeah, there are the hospitalization rates and the ICU rates are going down. So there's a couple of reasons for that. One is that lately we've seen that younger patients have been getting infected. So in the first wave, a lot of people with either underlying disease or people who are over 65 that are predisposed to worse outcomes were the ones going to the hospital and getting severe disease. So the rate of ICU went up. We also know how to treat this better now, right? So when we first started, you can imagine the doctors really didn't have a good handle on what is the best standard of care. Now we have drugs, we have different supportive care that really lessen the progression and severity of the disease. So not only are fewer people going on respirators, the case fatality rate, so the number of people who die as a percentage of people who get infected, is also decreasing. So we are doing better. It's still much higher than flu, the severe outcomes to coronavirus, but hopefully with the number of drugs coming online, you probably heard in the press, Lilly just got their antibody approved. We have remdesivir. We should have a couple of other drugs as well as a vaccine. We're hoping that case fatality rate plummets and this becomes a disease that we learn to live with like influenza.
Thank you, Dr. Chanda. That was great. So our next question is, why do young people not seem to be infected as often or as much as adults? So Dr. Renai, who would you like to answer that question?
Linda, you want to start with that one?
I can start. This is an interesting question because my husband is a pediatric infectious disease specialist, so he actually comes in contact with these patients who are not getting as severely infected as the adults. It's actually not completely clear at this time. They're absolutely getting the virus. That can be validated by testing, and they do testing when children come into the emergency room and into clinics and so forth. But I think that the jury is still out. And unfortunately, if we actually knew what was protecting the children, we might be able to capitalize on that so that we could better protect the adults. It's not intuitive that the children or the younger people would be better protected, especially young, very young children who don't really have the best immune system. And I think that most of us have gone through this period in our lives when our young children went to daycare and got every virus and were sick. So it's actually a surprising outcome. And there are studies that are being published. In fact, this whole field is very intense in terms of publications. So hopefully we will actually learn the answer to that question that will help inform us to our vaccine designs and so forth. Thank you.
That's great, Dr. Bradley. Thank you. So if 2 COVID vaccines from the FDA get approved, should we get both of them? That's our next question. So what do you say, Dr. Renai? Who should answer that one?
I'm tempted to challenge both Linda and Sumit on this. Sumit, you want to start? Linda, continue?
Yeah, so there's no reason to believe that getting multiple vaccines are going to be harmful, but I wouldn't take both of them at the same time. There's a way that actually you can tell if you're responding to the vaccine, and which is an antibody test. So this is the antibody test that people give to see if you've already had the coronavirus. So your body makes antibodies to coronavirus; those stick around. Those will also tell you if your body is responding to the vaccine. So what I would suggest is if you get the vaccine and your booster, wait 3 or 4 weeks, go get that antibody test from Quest or whoever your lab provider is. If you're making antibodies, you probably don't need to take another vaccine. If you're not, then I don't think there's any reason to believe that taking another vaccine would be harmful.
Thank you, Dr. Chanda. That was fascinating. I had no idea what you were going to say. Okay.
Either did I.
So the next question is, could getting the coronavirus make a person more susceptible to getting cancer? Dr. Renai, who would you like to answer that question?
So I'll start, and I would think that we need more time in order to assess and realize what would be the right answer, and time means perhaps even years. But overall, one would expect that if we are undergoing the storm immunologically and otherwise due to the infection, we may open the door for mishaps, and cancer can take advantage of that. I would appreciate a response from perhaps Sumit and Linda on this further.
Go ahead, Linda.
Can you just please repeat the question? Whether this virus can cause cancer? Is that what?
Yes. So is someone who gets COVID, are they more likely to get cancer?
So I would have to say that with Zev, we don't have enough information. However, there are definitely things that the SARS-CoV-2 virus does, which causes a cytokine storm, tissue damage, and other things, which could actually elicit an immune response that would be aberrant and would potentially be able to allow for an immune escape of cells. I think it's not so likely. And again, this would not be something that would show up very rapidly. If it happened at all, it would be something that would take place over a prolonged period of time. But just as viruses can escape an immune response, I think cancer cells can escape immune responses. So some of the mechanisms that are there for that to happen could potentially occur with any virus.
Okay, thank you very much, Dr. Bradley. So the next question is it true you can get COVID twice? If so, why? Who would you like to answer that, Dr. Renai?
The way I see it, it's rare and we really don't know enough. But Sumit, take it further.
Yeah, I mean, there have been reported incidents of people getting COVID twice, but it is, as Zev said, it's rare. And the likely underlying reason is that the first time you got COVID, your body did not mount a sufficient enough immune response for you to remember that you got the virus. And so, again, these are going to be the exception rather than the rule. If this was a common phenomenon, we would be hearing about more than the 2 patients. There was one in Hong Kong, one in Nevada, that were documented to get it. So I would say that for the most part, if you got COVID, and especially if you got severe disease, it's unlikely that you'll get it for at least the short term. We don't know how long that immunity lasts. We were talking about this before. And also, if you didn't get severe disease, it's more likely than not that your body didn't mount an immune response to the virus. So your immune system's not going to remember the virus, and so that would mean that you could get COVID twice.
Okay, thank you, Dr. Chanda. So the next question is directed to both Jenna Hope, Dr. Jenna Hope, and Dr. Sandra Leibel. So Did you ever imagine that you would be studying a virus in the middle of a pandemic? So that's your question.
No. I didn't think there was going to be a pandemic in my lifetime because, working in the clinical field and especially pediatrics, we vaccinate against everything.
Right.
So I think we've just gotten to the point where, there's a lot of parents that are a little concerned and are vaccine shy because we have such a short memory about all these diseases that we've eradicated that were so deadly in the past. And so, as a species, we kind of have this short-term memory, and we didn't realize that we do have to be proactive when it comes to these pandemic causing diseases. I'm very happy that I'm able to help in any way that I can, but obviously I wish it never happened. And I hope that we're better prepared for the obvious next one.
Sandra, you have helped, and thank you. So Jenna, do you want to answer that?
I mean, I agree completely with Sandra. Just, these are things that we've studied about, particularly if you've studied infectious disease, you studied the 1918 pandemic with influenza. So hopefully we can learn from this. And I mean, Sandra brought up a great point, especially about, vaccine shy new parents. I mean, we hear the words herd immunity in the media all the time, but the understanding, I'm not sure, is always conveyed, and that we need to maintain this level of protection if we want to keep these childhood diseases, or, in the case of SARS-CoV-2, not just childhood diseases, but if we want to keep those at bay, we definitely have to maintain that level of precaution. And I want to, make sure we get Laura's opinion too, because she's really working with the virus. I mean, that's a completely unique perspective.
Oh, thank you.
Wonderful. So the last question we have is, do the current clinical trials for vaccines include children? Does anybody, Dr. Renai, who would you like to handle that?
Linda, Sumit?
Yes, so at the moment, they do not include children.
Thank you. That was an easy, we got a yes/no question here. So I want to thank everyone here for being, I want to thank Linda, and I want to thank, Sandra, and I want to thank Linda and Jenna and Dr. Snyder and Sumit. I want to thank all of you for your time here today. And I want to thank the audience for being here. The Stanford Burnham Cancer Research Center appreciates you joining us. And we hope you've enjoyed it. And we hope next year, with all this work that's going on, we'll be able to hug each other again. So thank you very much. And it has been a pleasure to be here.